organization of the neural circuit and map the activity onto the wiring and connectivity of that neural system. And so just in the last few minutes, I want to maybe point a little bit to the future of where we hope to take these kinds of tools and to incorporate them into systems of ways of looking at neural circuits in a way that's inexpensive and deployable in individual neuroscience groups. And this last topic I want just to mention very briefly is something we call expansion microscopy. And we wanted a way that we could look at entire neural circuits, but get down to the resolution of individual synapses or axons or neural connections or even biomolecules. And so super resolution microscopy, of course, is very famous. Some of the inventors won the Nobel Prize a couple years ago for their efforts, for example. But it's difficult to apply to a large-scale 3D structure, like a brain circuit. It can be too slow, or it can struggle to image very deeply into it. And then electron microscopy gives you incredible resolution, but really struggles to get molecular information. So starting with two grad students, Faye Chen and Paul Tilburg, in my group, we decided could you actually take the brain and do the opposite, expand it and blow it up. And so this combines sort of two old lines of thinking. Toyuchi Tanaka was a physicist who worked out a lot of the physics of so-called ionic gels. These are polyelectrolytes, where, such as the sodium polyacrylate, maybe better known as the active ingredient in baby diapers. And when you add water, osmosis draws the water in, and you get this enormous swelling. And since the polymer backbones are highly charged, you get this even bigger expansion. And so he showed that these, the physics of this could actually nicely explain these 1,000-fold volumetric changes in the polymers used in the pure state. And the other thing you have to do is get the polymers inside the tissue, and Peter Hausen and Christine Breyer had worked out a protocol where you could take a specimen, like a tissue, and embed it in a hydrogel of polyacrylamide, back in 1981. And so we thought, well, are there ways that you could combine these kinds of strategies to embed tissues with polymers and then use soluble polymers in order to make it possible to visualize brain circuits? So if you do it just right, maybe you can pull apart the building blocks of a system like a brain and pull them apart into like a constellation of stars hovering in the sky. They would be there, suspended, but with their relative organization preserved. So to do that, we had to make a couple chemistries work. We had to develop anchoring molecules that could bind to DNA and RNA and proteins so they could be pulled apart. And we also developed ways of doing this with labels such as antibodies and other applied labels such as fluorescent proteins. Then we can form the polymers, and this uses a recipe that really combines the 1980 and 1981 papers I mentioned earlier. We're doing free radical polymerization that allows us to create these polymer threads throughout the tissue, and when the growing polymer threads encounter the anchor, they will bind. And that is sort of shown in this cartoon here. And then a third thing I had to figure out, which is also kind of tricky, is how to loosen up the molecules so that they lose their allegiance to each other and can be separated. So we use heat or enzymes or other methods to do that. And then we add water, just as Tanaka found many decades ago, the polymers swell, but this time the biomolecules or labels have come along for the ride. So here are some of the early examples of what we can find using this strategy. In panel B is a piece of mouse brain tissue before we expanded it. And then panel C is the same piece of mouse brain tissue a day or two later, where we added the polymer and then loosened up the molecules. In this case, we used proteinase K to chop up all the molecules that we didn't care about anymore, only retaining the fluorophores we wanted to finish later. And then panel C, you can see what happened. The brain is about 100 times bigger in volume, about 4.5 times bigger in each linear dimension. And the polymer mesh is very small. The spacing is only a couple nanometers, so one might hope that we could preserve the order of biomolecules down to a very fine scale. So here's the kind of data we can get with this strategy. This is the work we did with Dao and Kai, who's one of the people who worked on so-called rainbow labeling, where you express different amino epitopes in different brain cells, and then you come in with a fluorescent antibody and you can label them. So you can see here a piece of mouse hippocampus where the blue cell and the green cell have different kinds of epitopes. And if you zoom in, you get these beautiful pictures. And so the image at the top, you can see, I'll use the mouse, I guess, you can see what happens when you zoom in. It's a beautiful picture, but you lose sight of what these fine processes really look like. Here are, sort of green banana shape here, kind of hard to know what we're looking at. That's before you expand it. But the upper right is what happens after you expand. So the image, of course, is much bigger, but we shrank it to fit on the slide. And I hope you can see now that the green banana actually contains 4 individual axons running parallel to each other. And so we and others are now working on ways to use the ability to color code different parts of cells and also to color code different kinds of biomolecules to try to build up molecular pictures of how neural circuits are wired. You can use it for other kinds of biomolecules as well. This is a paper we recently did where Oz Rossi had joined our team, and we worked a lot with Alison Coate and Arden Rada's group as well. And we could anchor both proteins and nucleic acids that expand, and then image the location of individual messenger RNAs with nanoscale precision, even in intact brain tissues. And that's very interesting, of course, because neurons have one nucleus, but thousands of connections. Understanding how gene regulation is controlled at a spatial level might be very important for understanding learning and memory, disease progression, and other kinds of important functions and flaws in the nervous system. And then finally, how big can we expand? When J.B. Chong was a postdoc in my group, we worked on the idea of expanding something iteratively. So can you exponentially spatially amplify an object? So we took a brain, or a piece of brain tissue, we could polymerize it and expand it. Then we could form a second polymer in space opened up by the first and expand it again. So with 2 steps, you could have 4.5 times 4.5, which is about 20-fold linear expansion. And so this is very exciting to us because now we and others have characterized this protocol, and we think that we might be having a resolution now that is equal to or comparable to any other nanoscale resolution imaging strategy out there using light microscopy. So here's some examples again using rainbow. In the upper left, we have unexpanded. It's beautiful and hard to see. We have the right, with one round of expansion, we can see the axons and dendrites, but we are missing out on some of the features of fine compartments like synapses. And then the lower left, you can see what happens with double expansion. So we can get down to the finest compartments of cells, such as the spine necks, that are difficult to see in neural circuits. And if you look carefully, the lower left image looks kind of dotty, looks kind of punctate. And so we're excited about the idea that we might be approaching the resolution of single biomolecules. And so we're working now to validate this by expanding lots of different objects that have known properties, such as viruses and other kinds of ground truth understandable biological systems where the components are well known and well understood. Expansion is really taking off. We've seen it applied to all sorts of different critters, bacteria in the upper left, Giardia parasites in the lower left, and right now the kinds of discoveries people are making are coming along at a rapid clip. So just to wrap up, we really are excited about this idea of nanoscale imaging that can be done in a highly scalable way, and also that can be done with existing hardware so that people can use these tools. A big theme of what we do is to try to think of tools that are easily deployable into the field. A lot of that depends upon performance, but also ease of use. And a theme of all the different technologies that we try to explore is indeed that biology, which is a very democratized kind of endeavor, should benefit broadly from these kinds of technologies. So internally in our group, we're very excited about synthesizing these tools together. Could you watch and perturb in real time in interesting behavior the neural activity in a neural circuit in a rate-behaving animal? And then at the end of the day, could you map out the connectivity of the cells and the molecules at those connections in order to synthesize emergent models of how brain circuits work? But also, we're very excited about the process of teaching as well, and we share all of our technologies freely, and we, for visitors who come to our group, we welcome people, essentially every week to come by and hang out and watch us do these kinds of experiments. And so we're always eager to teach and help people use these tools. So I think I'll end there. I think one of the themes of the morning is that collaboration is key. In the case of our group, it's been a pretty fun roller coaster ride with a lot of different disciplines, from physics to chemistry to ecology, colliding in interesting ways. And I'd like to end again by expressing my honor and gratitude for being recognized here, and also to thank Peter and Carl for all the great collegiality and collaboration over the years. So, thank you very much.